Hi, in this video we'll look at some of the mechanisms of oxidative phosphorylation. We'll take a closer look at the proton gradient across the intermitochondrial membrane, showing that it's an electrochemical gradient with both electrical and chemical concentration components that contribute to the driving force that drives the synthesis of ATP. We'll look at the protein complex, the ATP synthase, which is responsible for ATP synthesis, and we'll show that it's a rotary powered machine, and rotary motion is a fairly rare phenomenon inside biology. We'll look at the numerical accounting of ATP produced, and we'll show that different sources report different numbers. There's a consensus of approximately 36 ATP produced for each molecule of glucose metabolized. However, we'll accept anywhere between 30 and 40 ATP for a molecule of glucose. And lastly, we'll look at proton pumping in aqueous-based systems. There's some unique features of pumping protons that are not valid for any other ion like calcium, potassium, or sodium. So in this slide, we look at the electrochemical gradient across the intermitochondrial membrane. We'll start here at the bottom, which highlights the concentration difference of protons in the intermembrane space versus the intermitochondrial matrix. The diffusion of a proton from a place of high concentration to low concentration is energetically downhill. But additionally, when we consider the electrostatics, as in up here, because you have more protons out here and they're positively charged, you have positive charge repulsion on this side of the membrane, and you have an attraction of protons for the more negative electrochemical potential on the intermitochondrial matrix, so that the transfer of any proton across the membrane in this direction is energetically downhill because opposite charges attract. The equation called the Nernst equation is used to calculate the relative contributions of the two forces, and we'll discuss this in class. On this slide, we see a realistic depiction of the ATP synthase, also referred to as the F0F1 ATPase. This protein complex sits in the intermitochondrial membrane. This is the intermitochondrial matrix side, and this is the intermembrane space between the two membranes. Again, the proton gradient, there's excess of protons on this side of the membrane, and they will flow through channels between the A and the C protein subunits. It will cause this red stalk to rotate in a direction, I'm not sure clockwise or counterclockwise. The alpha and beta subunits are stationary, but because the inside stalk is rotating, the alpha and beta subunits will change conformation, and that will cause ADP to be converted to ATP. Here you can see part of the crystal structure of the stalk penetrating into the alpha and beta subunits. And if you want to see a great video, a short one, 2 or 3 minutes, of the F0F1 ATPase in action, go follow this YouTube link, which is linked to on the class home website. There is one other component in biology that involves rotary motion, and it's very similar, and that's the rotation of flagella, which are tails that can propel bacteria in a swimming motion, which works on sort of the same principle of a proton gradient in a small space which diffuses through a channel protein, causing conformational changes in rotor proteins, which will induce a rotational motion. Now in this slide, we start to delve into some of the discrepancies on the number of ATP produced per glucose molecule. This is actually an older version of Wikipedia, but this was the table that was contained, and this is sort of the most common accounting, at least until 5 or 10 years ago, where lower numbers started to take hold. But just to run through it, you'll remember that in glycolysis, the first thing we did is we used up 2 ATP to create phosphofructase 1,6 fructose 1,6-biphosphate, but then we get 4 back of substrate phosphorylation during steps 6 and 7. We also produce 2 NADH, which can produce as many as 3 molecules of ATP per NADH, but the NADH, which is produced in the cytosol, has to get transported into the mitochondria, and then depending on the concentrations of other reactants, you may have to expend some energy transporting NADH itself. Once inside the intermitochondrial matrix and going through the enzymatic processes involved with conversion of pyruvate into acetyl-CoA and the citric acid cycle, so you most often get 3 ATP per NADH molecule. These 2 molecules of substrate level phosphorylation inside the intermitochondrial matrix are the 2 molecules of GTP, and then we also produce, since we go around the Krebs cycle twice, we get 2 molecules of FADH2, and you get 2 ATP per FADH2. We will not memorize that for this course, but you will be expected to know that the processes requiring oxygen inside the mitochondria produce the great bulk of ATP, including those from the NADH, whereas anaerobic respiration occurring in the cytosol produces only a small net of 2 ATP. If you're taking the MCATs, you do need to memorize this distribution of ATP molecules. However, on this slide, taken from your book, we'll see that they're less generous on the number of ATP produced by NADH. Again, Wikipedia had 18 for these 6 NADH. So I think you can think of 38 as being a maximum and 30 being more of a typical value that's obtained by experimental measurements, whereas where the costs are assumed to be greater for transporting molecules to the places they need to be to get the ATP produced, such as pyruvate, NADH, etc. Again, we'll accept any value between 30 and 40. So in this slide, we'll start to discuss proton pumping in aqueous environments. And the punchline is going to be that although it appears a proton is moving from, in this case, this location to this location, what's really occurring is a rapid reorganization of hydrogen bonding, which can occur much more rapidly than actual physical transfer of an atom. If you look carefully at each of these 5 drawings, the position of the protons don't really change. Let's look at this proton. Here we go. Here we go, here we go, here we go. But the impression is given that because of change in hydrogen bonding and where the actual bonds are, that a proton is moving. Again, here we have a positive charge on this water molecule. Here we have the positive charge on this water molecule. Here we have finally the positive charge on this water molecule. It seems like a proton has transferred from this oxygen atom to this oxygen atom. But what has occurred, again, as we see, none of the protons have actually changed position. It's just that the bonding has changed within the hydrogen bonded network. I suppose I should also say that when people write about H plus in solution, they're really talking about H3O plus as an altered form of water. There's no such thing, and again, sorry that this is messy, there's no such thing as free protons in an aqueous solution. So hopefully this will be clear what we mean about proton pumping in water. When you remember that in proteins there's an extensive hydrogen bonded network, you can at least conceptually anticipate that the same sort of network alteration of bonds and hydrogen bonding can occur to apparently cause a proton on one side of a protein all of a sudden appear at the other end. It's not actually the same proton, but the net effect is of taking up protons from one end of the protein, to the other end. Now in this slide we want to show the coupling between electron transfer and proton pumping. In this example, proton B is the protein that pumps the hydrogen ions, the protons, and once an electron is transferred from protein A to protein B, now you all of a sudden have a new negative charge on protein B, and so that for electroneutrality, or you might find it quite reasonable that a positively charged proton from the aqueous solution would be added to an ionizable group here in the protein. Now that you've picked up an electron and a proton, you've changed the chemical composition of the structure, and it's very reasonable to have a conformational change of that protein. That conformational change might trigger the electron transfer from protein B to protein C, and now you would have an excess positive charge which can be lost to the aqueous environment. The sleight of hand, if you will, is that the proton being released is not physically the same proton that was acquired during the initial electron transfer reaction. Had evolution not been designing a protein whose function it is to pump protons after the electron transfer reaction, it may have very well given up its proton back to the same side of the membrane. It's just that in this scenario, because of the conformational change, the most likely proton to be lost is lost to this side. Now the proton that was lost here will be replenished by the hydrogen bonded network that exists within the protein, and that when this one's picked up, it'll cause all the protons to shift one unit towards this other end. But the take-home point is this proton that's being released is not the same proton that's being picked up. And on this slide, I'm going to give you a break because I've rambled, but also because I don't like this picture so much as a mechanism for proton pumping. Again, it's kind of going against the point I've been trying to make. This makes it look like it is exactly the same proton that is being released on the other side. I'm going to disagree with this slide. So in this last slide, I want to talk a little bit about the electron transfer reactions. And as you can see here, we have a redox energy scale, with the electrons from NADH being very high energy. They're being transferred to first the NADH dehydrogenase complex, to the ubiquinone, to the cytochrome BC1 complex, to cytochrome C, to cytochrome C oxidase, and finally to oxygen. That each of the electron transfers is energetically downhill. Each time the electron is transported to one of the three primary protein complexes, a proton is pumped across the intermitochondrial membrane. Now you might wonder what prevents an electron being transferred from the NADH dehydrogenase complex directly to cytochrome C oxidase. And at the risk of confusing people somewhat, the rate of electron transfers are finally tuned rather acutely to the energy drop for an electron transfer. In this case, electrons transferred between these two molecules and these two molecules. And it actually is the case that a very large electron energy drop, let's say from protein complex 1 down to protein complex 3, actually occurs at a much slower rate than sort of an optimally tuned energy gap. That's beyond the scope of this course, but I think it's a question that many of you might puzzle about if you spend a little time thinking about this. We'll discuss a little bit more about the energy gap of electrons in oxygen and water and molecules that you might use to synthesize molecules like NADH or NADPH when we discuss photosynthesis in the next couple of lectures. Thanks for listening.